Are we recording? Last class. Go. We had just sort of finished up or started practicing our incomplete dominance. And so you should have brought a completed practice question number 2 with you. I don't want to go through the whole thing, but I do want to focus very tightly on part C. So let's look at this as if we've been given this as a test question. If I was given this as a test question, how would I know that this was incomplete dominance, right? So when I'm looking at this, right, I'm looking at the list of phenotypes that I've been given. And I note that I have 1, 2, 3 different phenotypes. And the fact that I've been listed 3 phenotypes is already a good flag for me that this is incomplete dominance. Right, that was one of our defining features. The other thing that I would use here is as I actually look in detail at what these phenotypes are, I see that I have black, white, and gray as this last one. Okay, so remember when we were looking at the definition of incomplete, okay, we said that that third one was a mixed or blended phenotype of the 2 dominants. Okay, and so that's what we really see here, right? If I look at gray, gray is really an intermediate or a blend between black and white, right? I can't go into gray and circle chunks of black. I can't circle chunks of white. Okay? If I think of this as paint, which is a good way to think of this, right? I know that I put black in there. I know that I put white in there. It's been mixed up, and now it's become a brand new, unique thing. Okay, somewhere in between. So those 2 parts together, the fact that my third phenotype is a blend, and the fact that I had 3 phenos together, is a really strong indicator to me that this is incomplete dominance. I have too many phenotypes for this to be simple dominance, right? That only ever had 2. Okay, so let's work through this together then. All right, now I'm just going to work through part C. So I have a gray-eyed alien and her black-eyed partner. So first, let's build a legend together. All right, so based on the question, I know that gray is my blend. In other words, that's my heterozygote, right? So I know that black is going to be homozygous. I'm going to give that a dominant letter. Right, remember, incomplete dominance is 2 dominant yelling at each other. I'm going to use capital B for black. You did not have to. You want to compare this to the work that you did. Okay, so then I'm going to look at white. I know white is my other dominant. I'm going to use a capital W for that. Okay, remember, this is also dominant. We think about this as 2 people screaming at the chip bowl, and their stories get mixed up or blended together. Like our kangaroo in Africa. Okay, so now let's look at our cross. I have a lady gray-eyed alien. And now we said gray was being blended. So I know that that's my heterozygote. So I need 1 black allele and 1 white allele to do that. Okay, now her black-eyed partner, which for the sake of his simplicity I'm assigning as male. I don't know how this alien species rolls. If I'm going to be black-eyed, I have to be homozygous for the black allele. I have two B's. All right, so if we do gametes, remember, we want these to be haploid gametes. So each should have one allele of each letter type. The dad can make two sperm. The first one will have a single bee in it for the first bee. The second sperm will have a single bee in it for the second bee. Mom is making eggs. And she can make a B and a W. All right, let's make up a Punnett square. Remember, we always put the ladies across the top, males along the side. Okay, and above or next to each box, right, we always put one gamete. There's my Punnett square. And then we do fertilization to make our potential babies. So, can our alien couple have a white-eyed baby? No. Why? So in this case, there's no offspring that have two W's. Okay, so we're 50% black-eyed, 50% gray-eyed. Okay, what would we have to do to make a white-eyed baby? What has to change here? Both parents? Which parent? Well, what we've assigned as male here, right? Right, so the one that has black eyes. Right, if we want a white-eyed baby, so if I'm a white-eyed baby, okay, that means I have two W alleles. I'm homozygous dominant for W. And those two alleles, I got one from mom. And mom can give me one, right? She can. But then I also got one from dad. Okay? And as this is set up, dad can't give me a W allele. Okay, so I would need a parent that can give me a W allele. Good. Any questions about that? Okay. Any other questions about incomplete dominance or some of these other questions that you should absolutely be doing? Because you came here with a totally complete packet, which is not on your phones. Genotypic ratios? Okay, so let's do it for this question. And not in yellow, because that's horrifying. Okay, so if we're doing the genotypic ratios, and I'm just going to do it for this last question. That kind of makes sense anyway. So remember, the first thing I want to do is list all of the genotypes that I have. So in this case, I only ended up with 2. And then next to that, we want to list our box count. So I have 2 of the black and 2 of the BWs. Or remember, we can reduce these if we want to, right? Because 2 to 2 is equal to 1 to 1, like dealing with fractions. Okay. Both of those are equally acceptable. Okay, now we're going to do the same thing. We would translate our boxes. Okay, so what I would probably do is go through and add translations to all of my boxes. That makes my life a lot easier. Then I'm going to list them here as well. As well as the ratios that we have. Now, if you did this for part B, you may have a different answer. Or if you did them for both, all right, that's fine. Just make sure you're clear. Did everybody see what I did there? Did I answer your question? Good question. Okay, so the big key when we're doing our ratios is just make sure that we're clear. Because all it's really saying is what is the likelihood of all of the different types of potential babies that we've put forth here. So here we're just saying you have an equal likelihood of having a black-eyed baby and a gray-eyed baby. 50-50. Any other questions about incomplete dominance? Again. Okay, so remember the key here when we're doing incomplete dominance is our 2 defining features. So we get 3 phenotypes, okay, and this third phenotype is blended or mixed. And that we still only have the 2 alleles, just like we did in simple dominance. Okay, but in this case, they're both dominant, right? So instead of having a dominant and recessive, here we had dominant and dominant. Let's compare that to our next one, okay, called codominance. Now there's a lot of things here that are going to feel similar. In fact, this is some of the easiest to confuse, codominance and incomplete dominance. We want to focus on how can I tell the 2 apart. Okay. So we see right off the bat, we have something that's similar. Okay, in incomplete dominance, we had more than one dominant gene, right? More than one person yelling at the table. And in this one, we have more than one dominant gene, right? More than one yelling at the chip table. Okay, but here are some key features that are different. So here are our 2 dominants. one, okay, we're still going to retain recessive genes or alleles. And so here's a recessive allele. Okay, so at this party, in addition to having the individuals yelling at the chip bowl, okay, we still had people talking to cats. It's a wild party. So if you see this situation, right, we know it can't be incomplete dominance, right? Because incomplete dominance didn't have any recessive alleles. The other thing that's really different here is if we look at how our dominant heterozygotes look. Okay, so remember with incomplete dominance, the key was you got that real blended effect. It's like mix and paint. And with here, if we look at our heterozygote with our dominance, it's not so much blended anymore. It looks like a tossed salad. Okay, the stuff's still mixed together. Okay, but sort of like when you have a mixed or tossed salad, right? I can still go in and I can pull out, this is a piece of lettuce, right? This is a crouton. This is a piece of shredded carrot. And they're all in the bowl together. Okay, but each piece is still individual and clear and obvious. So we don't have any unique blending. Okay, so what I usually say to do is go through and say, if I have this, I can take my homozygous dominant. I see this phenotype. Can I go into my heterozygote and pull it out like pieces of lettuce? In this case, I can, right? There it is. There it is. I can go through here and I can nitpick this out. I can circle those in a way I could not do before. I couldn't go out and circle parts of black. They weren't there. Okay, but I can do that with all of my parts here. I can pick them out. Pull the tomatoes out of my salad. Okay, if I can do that, if I've got a salad, that's codominance. Okay, so the heterozygous mix here looks a lot different. Okay, we're still retaining some of that individuality in that final phenotype. Also, when we're looking at this, how many phenotypes do I have? 4. How many did I have in incomplete dominance? 3. Right? Red, pink, and white. For example, right, from our last slide. Okay, and that's because we have that recessive in there. Right? Our recessive gets a phenotype too. Let's not forget the poor guy. So we're also going to have more phenotypes usually. We can use all of these things to help us realize the difference between incomplete and codominance. That's going to help us a lot. Any questions about the definition? Let's look at one. Unfortunately, my sheet is a little mucked up a bit. So we're actually going to skip to 4, and we're actually going to do 4 twice, because I'm going to simplify it here. So, we're going to do 4, but you're going to ignore all of the plus and minus business right now, because that's going to make it polygenic, and we are not ready to deal with that. The question works just fine if we just deal with the A and the B. Let's set up the legend together. So remember from our last page that you fundamentally can have 3 blood types, right? I can be A, I can be B, and I can be O. And I can, of course, be AB, right, if I put my heterozygotes together. So we know that O is our recessive. And we learned that from the previous slide. Okay, so we're going to give our O a little I. This is the actual notation that we use when blood typing happens. So might as well use it. So if I'm going to end up with an O phenotype, right, I need 2 little i's. 2 quiet little guys talking in the corner of the cat. That's the only way we can hear them. All right, now here's the deal, and so this is going to look funny. We need practice working on this. When we're dealing with these sorts of things, we're going to see this notation a handful of times. I have a recessive gene that's a little I. If I'm going to have a dominant allele, I have to use the capital letter for that, right? The problem is, if I just do capital I, now I can't tell them apart. Okay, so I want to tell them apart, but know that they're still associated with our little recessive friends. So I'm going to add what's called a superscript. It's kind of like adding an asterisk to it to tell them apart. I'm going to add a little b to this one and a little a to this one. Okay. It's important to note this does not make it 2 letters long. Okay, this is still just 1 letter. Okay, so we're not going to split this up. Okay, this still is haploid. Okay, this is all one thing. Okay, like I said, it's just like adding an asterisk to it so we can tell them apart. Okay, so you have I asterisk A. It tells me that's the dominant letter for blood type A. I asterisk B tells me it's the dominant letter for blood type B. And little I, okay, the recessive allele for blood type O. So this is weird notation. This is brand new for us. So using that notation and the information in the question, okay, go ahead and make a cross for us. what do we got for our cross here? So we know mom is type A. So what is her genotype going to have to be? Is that the only option? So this is really common with blood types or with a lot of things, right? So remember, now that we're dealing with that recessive again, we are dealing with uncertainty again. So mom can absolutely be homozygous dominant. That would give her type A. But all we really know is that for sure she has at least one dominant A. Because the one after it, her second one, could be a recessive and just be quiet or covered. And we're going to have the same problem with dad, right? He also has to have at least one, in this case of the B. But he may also have the heterozygous with the recessive, right? So it's just going to be covered and quiet. So I would pick one of each when you set this up, if you haven't already. Okay, let's keep working through my example. Keep in mind that depending on what parents you chose, there will be some variation in the answers to your questions here. Because you can have up to 4 different Punnett squares. All right, so given what I have, will we have any children that are universal donors, defined here as type O blood? Will not. Correct. Why? I don't have any babies that are little I, little I. Okay. How might I have changed that so I did? Or how would that happen here? Excellent, right? Both parents would have to be able to donate a recessive allele. In my particular setup, only one of my parents is donating a recessive allele, right? My mother I set up as homozygous dominant. Okay. So none of my boxes can be recessive I, aka blood type O. Right. Remember, if I think about if I'm a child, okay, so my child is going to be whoop, whoop, little i's. That yellow is really atrocious. Okay, I got one I from my mom and one I from my dad. I can get an I from my dad here, but I cannot from my mom. But the way I have this set up. Now, depending on what parents you chose, you may be able to do so. Now let's look at the second question. Will any children have blood type like their mother? Yes, in my particular case, right? Well, their phenotype at least. Go ahead and do my boxes out. What blood type are those? AB, right? That's not helpful. What about those? What are those? Blood type A. Right? Now, it's not the same genotype as mom, but it is the same phenotype as mom. And again, your actual answer here may differ depending on what mom and or dad that you chose. Okay. Same with these. These will vary greatly, right, depending on what parents you chose. We're still going to handle them the same. Right, list what's in your boxes and then tell me how many of each box you have. And here in particular, you will see that despite the fact many of our previous ones, the genotype and phenotypic ratios have had the same counts, this one will likely not. Mine does here, but yours will not necessarily. Okay, so using the information I have, right, I have recessive alleles, okay, multiple phenotypes. I know that this is dominant. Any questions about codominance or the difference or definitions of codominance versus incomplete dominance? That's a really big one we want to make sure we understand. Does this feel okay? Blood type is a really common example of codominance. We're going to go through another related one here in a few slides to help give you more examples of how to pick this up. Okay. Next relationship. Okay, in this case, we're not looking for differences in genetics per se, but how the DNA is actually expressed. We call this idea plasticity. Plasticity is another word for flexibility. of have to mold our head around that since we tend to think of the word plastic as being hard. Okay? We want to, in this case, think about it being as moldable or flexible. So in this case, phenotypic plasticity is referring to how genes are turned on and off based on the environment that they're in. Okay, so if I'm in a warm environment, okay, that warm environment will have my genes make specific proteins, okay, that help me live in a warm environment. Okay, if I'm in a cold environment, okay, that cold environment is going to have my genes make proteins that help me survive in a cold environment. Okay. So even if I have the same DNA, being in one or the other of those environments is going to alter what proteins my DNA makes, and ultimately how my body reacts, which is ultimately how I look, right? So another way we can think about this, it's a little easier to imagine, is the twin effect. So we've all experienced this at one point in our life. Okay, so most of us at some stage or another know a set of identical twins. It has to be identical, not fraternal. Some of you are secretly identical twins. That's always fun when I find this one out. But so think about that set of twins that you know. Okay? And think about them a long time ago, okay, as early back as you can remember them, kindergarten. Okay, so when you first met them, likely, whether you like to openly admit it or not, it was kind of hard to tell them apart, right? They looked super similar. A lot of parents joke about this. When they first had their twins, they had to make sure they always dressed them in the same colors, make sure that they didn't mix them up, because they look super similar, and they should, right? They have the exact same DNA. And so when you first met your twin friends, or friend, depending how you are, okay, in kindergarten, right, or as early as you can remember, they probably look super similar. Not identical, certainly, okay, but very, very similar, because they'd had the same experiences probably, right? They'd always lived in their parents' houses. They'd been fed the same meals. They were allowed the same outside time. Okay? They went to the same summer camps, probably. You're looking at age 6 here. You don't delineate a lot from your siblings at age 6. Okay? But think about how your twin friends looked then compared to now. Okay? You probably can tell them apart now, no problem. And it's not just because you got to know them, which is part of it, but because they do distinctly look different now. Okay? Because they've grown up and they've gotten to be their own people now. Okay? They got to decide how much they were going to eat to dinner. Okay? Whether they were going to play sports or be in the band. How much time they were going to spend outside. Whether they were going to make very inappropriate decisions like to smoke or drink. Okay? All of these things matter. And it's had an effect on how much they've wrinkled, what color their skin has turned, if they've gained weight or not. I have twin aunts, so much older for me, right, because they're older than I am, and I know you all think I'm fabulously young, but they're certainly older than I am. And despite the fact that one does insist on, like, feathering her hair like Heather Locklear from 1982, which does make it very obvious, they don't look a ton alike. Okay? One has been smoking since she was like 13, which is a super weird fact that she likes to tell people. And that has an effect, okay, on the way her mouth and face looks. Okay, but they have both made largely different decisions, even though they still live together, which is also super weird. But they have spent different times outside. One played sports for a really long time and the other one didn't. One liked to tan and the other one didn't. Right? So all of these things made them look really different. And you can tell they're twins. Right? It's there in their facial structure and in their eyes, like very obviously. But not for a second would you confuse them. Right? Here in their late 40s, early 50s, I should probably know that and I don't. So the older they get, the more decisions they make, the more different they look. And so this is what we're talking about with phenotypic plasticity. So if we look at our bunnies here, this is what's happening. Let's look down here at our black and pink bunnies. So these bunnies are also twins. So cute. Not like my ass. Right, so we took our little black bunny, and when he was just born, we put him in a little cold cage. We took our pink bunny, and when he was just born, we put him in a little warm cage. Okay, so despite the fact that they have the same DNA, okay, depending on the environment these bunnies were in, their DNA coded different proteins in response to the environment that they were in. So in the cold cage, our little black bunny, responding to that cold environment, created proteins to darken the melanin in his skin. Kind of like polar bears. They have white fur that helps them camouflage, but the skin under their fur, like many other Arctic animals, is black. It just helps them absorb sunlight and get warm. Our pink bunny, who was in a warm cage the whole time, did not need to do that. Okay? And so did not activate the proteins in his DNA to create darker skin. Okay, so this is what we're talking about when we're talking about plasticity. And they have the same DNA. We don't have different alleles or different genes or any of that business. It's just the effect of having different environments activate different proteins that are already there. So we're not going to have a practice problem with this one, right? Because all of the DNA and all of the alleles would be the same. It would be rather boring. The key here is, and this is what we're talking about with this one, right? The environment is changing what is being coded or what's being turned on and off. Okay. Honestly, I'm not going to start this. I just want to have something for you guys to put in. Your teacher's question. This is way too big of a topic to start in a minute.